Good afternoon, everyone. I'm George Kunos, Scientific Director of the Alcohol Institute, and it is my pleasure to introduce today's WALS speaker, Dr. Amita Sehgal, whose lecture is co-sponsored by our institute and the Sleep Biology Interest Group. Dr. Sehgal received her undergraduate education in her native India. She then moved to Cornell University for doctoral training in the laboratory of Moses Chao, where she studied the biology of nerve growth factor and eventually cloned its receptor. Having earned her Ph.D. in cell biology and genetics, she then moved to the Rockefeller University to do postdoctoral training in the laboratory of Michael Young. That is where her enduring interest and work on circadian mechanisms started, and continued in her own laboratory at the University of Pennsylvania, where she's currently the John Herr Musser Professor of Neuroscience, a Howard Hughes Investigator since 1997, and Director of the Chronobiology program. Starting with the amazingly simple but very important discovery that fruit flies actually sleep, Dr. Sehgal used the Drosophila as a model system to dissect the molecular mechanisms underlying circadian rhythms and its most conspicuous form, sleep. Her laboratory has been able to identify several genes and their protein products involved in the control of circadian behavior and sleep, including the timeless clock gene, the jet lag gene and its protein product, which control the circadian degradation of timeless, the sleepless gene and protein, loss of which make flies sleep. I find it really endearing that all of these genes have very common sense names, easy to remember, and the red-eye gene, whose expression correlates with wakefulness. She also discovered the morphological substrate of the control of sleep, the mushroom bodies, a specific brain structure in the fly, which plays a key role in the control of sleep, along with other important biological functions. Her interests go beyond the molecular basis of sleep and extend to understanding how circadian and sleep systems interact with other important functions, such as metabolism and reproductive behavior. Dr. Sehgal has consistently published her finding in top-tier journals, including Science, Nature, Cell, and Neuron, and has received numerous awards and honors, including elected membership in the American Academy of Arts and Sciences and in the Institute of Medicine, also a Javits Award from NINDS, which is analogous to a Merit Award, and a Senior Scholar Award from the Ellison Medical Foundation. Before yielding the podium to Dr. Sehgal, I'd like to remind everyone that following the lecture and the question and answer period, there will be a reception across the hall here that everybody present is welcome to join. Thank you, and Dr. Sehgal, we're all excited to hear about your work.
Now can you hear me? Yes. Okay. All right. Thank you very much, George, for that very nice introduction. I'm really honored to be here for this lecture series, and a big thanks to Matthew Pava for hosting me and planning my itinerary. I've had a really great day connecting with friends and colleagues, some new, some old, including former trainees from my lab. So I'm really delighted to be here. So, what I'm going to tell you about today is, yep, it works, okay, the work that we're doing in my lab to understand sleep. Now, sleep is a behavioral state that's seen in organisms, many different species, some of which are shown here on this slide. We, for most of our studies, not entirely exclusively, but for most of our studies, we use the fruit fly Drosophila melanogaster to understand sleep. And what I'd like to do today is sort of give you a flavor for how we're using the fly to address different questions that we all, or many of us, have about sleep. And these questions are basically relates to, why sleep occurs, when sleep occurs, so the daily timing of the sleep-wake cycle, which is, of course, controlled by the circadian clock. why sleep occurs. In other words, what is the function of sleep? Why do we need to be in this state? And then finally, what is it that regulates sleep onset and the maintenance of sleep? So you don't just go to sleep because it's a specific time, right? It's because the clock is telling you to go to sleep. You do it because you're actually sleepy. So what is it that brings on that sleep onset and maintains it? And I should point out that a lot of times you'll be sleepy at times when your clock might be telling you to wake up. So it doesn't always go with the clock. This really speaks to the homeostatic regulation of sleep, and I'm calling this here how sleep occurs. So to begin with the daily timing of sleep, so what you're looking at here is Earth's day-night cycle seen from the moon, basically just to stress that we live in a cyclic environment. And to adapt to this cyclic environment, organisms have evolved endogenous timekeeping mechanisms, which time behavior and physiology such that most aspects of these occur with a circadian rhythm, a 24-hour rhythm. And these are but a few of the processes that in humans and in most other species show circadian regulation on some level, right? So metabolism, for instance, shows circadian regulation on almost every level. Cholesterol metabolism cycles, glucose metabolism cycles. But for purposes of this talk, what's important is the circadian timing, or like, as I called it, the daily timing of sleep. And as I said, we use Drosophila for this question. And so how do we measure sleep in Drosophila? So we place individual flies in glass tubes, such as the one shown here. When the fly is active, it marches back and forth in the tube and deflects an infrared beam that's projected into the tube. The fly can't see infrared light. And so this crossing generates an event picked up by a computer. And after a week or so of monitoring, you get an activity record that looks like this, where the shaded areas correspond to activity. Now, for many years, we had actually been studying rest activity in the fly as a readout for internal clock function. And we weren't particularly interested in what, the rest periods here were. But at some point, as George pointed out, we became interested in what the fly is doing when it's resting, and we realized that it really is in a sleep-like state. If you're interested, I can tell you later why we decided it was a sleep-like state. Suffice it to say right now that this model, which was developed simultaneously by Paul Shaw and Giulio Tononi's lab, has now been accepted worldwide, and so there are dozens of labs studying fly sleep. So how do we get sleep data out of this? So we actually determine sleep from this as every 5-minute or more period of immobility. And again, I can, in the interest of time, I won't go into how we came up with that 5-minute definition, but this is the standard accepted definition for sleep in flies. It's a 5-minute or more period of immobility. So the sleep record from data from a record like this would look something like that, where this is daytime sleep, this is nighttime sleep. Flies are diurnal like humans. Most of the sleep occurs at night, but they do have an afternoon siesta. All right, so how do you get this timing of the circadian timing of the sleep-wake cycle? Now, it turns out the fly has really been a great system to study circadian rhythms, the molecular basis of circadian rhythms. Oops, I went backwards, sorry. Okay. And also the cellular basis of circadian rhythms. So I'm not going to dwell on the molecular details of the clock today, but more so on the cellular basis and on the circuits that drive circadian rhythms. So what you're looking at here is the fly brain. And here are the different groups of neurons in the fly brain that express circadian clock genes. So these then are the clock cells. And they're all bilaterally represented, shown here on one side. So there are these 6 different groups. And of these different groups, the one that's circled here, the S, L, and Vs, are the ones that are most important. So a clock in these neurons is necessary and sufficient to drive the circadian rhythm. Now, it's not to say the other groups don't contribute, so it turns out that different aspects of the behavioral rhythm, like the morning peak of activity and the evening peak of activity, are driven by different groups of neurons. But these are the most important. But it's clear that we're gaining like a lot of understanding about the molecular clock and about the cells that are part of the clock system. So the aspect of circadian biology that is least understood in Drosophila or in any organism for that matter is how signals are carried away from the clock cells, these circled here, to produce rhythmic behavior. Okay, when you're looking at a metabolic function of the clock might directly affect transcription of a metabolic gene and make it cycle at the level of the RNA. But when you're looking at a behavior, you have a circuit involved. So the question is, what is the circuit, the output circuit that conducts signals away from these neurons and produces rhythmic behavior? So what are the output circuits? So what we knew about this when we got started on this project was that these cells extend to the dorsal part of the flybrain. This is dorsal, this is ventral, and they secrete a neuropeptide called pigment dispersing factor. Now, the person in my lab who did this work is a former postdoc who has just recently started his own lab, Dan Cavanaugh. And what Dan did was to try to identify neurons of this output circuit. And the approach that he took was one that's actually quite popular in flies, which is to use an unbiased method to determine which neurons are required. And the unbiased method consists of perturbing the electrical activity of random subsets of neurons in the flybrain. So the rationale is that the neurons that are part of the output circuit for rhythmic rest activity or rhythmic sleep-wake are going to have their electrical activity regulated. So if you constantly activate them, the behavior will become arrhythmic. So Dan conducted an unbiased screen for such neurons that are required, and I'll just summarize those findings here and tell you that he found out of 110 lines that he screened, which targeted different subsets of neurons in the flybrain, he found 10 that were required for this rhythmic sleep-wake. And now you'll notice that, so this is the expression pattern of those different lines. So these are the parts of the flybrain they're expressed in. So it's a GFP reporter. And you'll notice that the GFP in many of these is quite widespread. Some the labeling is a little more sparse than the others. But what we noticed was that the region of overlap in all of these was this part of the dorsal flybrain. And so all of them labeled this part here, which is shown in the inset. And that part of the flybrain is a region called the pars intercerebralis. And the pars intercerebralis is a group of peptidergic neurons that is thought to be equivalent to the mammalian hypothalamus. In flies, prior to Dan's work, it was best known for being the site of the insulin-producing cells in the fly. So insulin in the fly is produced in the brain, and flies don't have a pancreas. Okay, so this region is important, and this region does not itself contain a clock, so it's downstream of the clock cells. So we asked then whether these clock cells connect to this region, and so Dan did some anatomical tracing to figure out whether the clock neurons connect directly to this region, and he found that, in fact, they do not. Okay, he used a technique called GRASP, where you look for GFP reconstitution across synaptic partners as a way of mapping the synaptic connectivity. And he found that these neurons do not connect here. Now, I should point out, too, I've been asked this question before, these neurons are secreting a peptide. So could this peptide then just not diffuse across to this region? Do you have to have a synaptic connection? And the answer is that for this particular peptide, the diffusion is limited. So yes, you do. Okay, so what Dan found, though, was that this other group of clock neurons in the dorsal part of the flybrain, the DN1s, they connect directly to the PI, and the central clock cells connect to the DN1s. So there is this polysynaptic circuit from these central clock cells to this putative hypothalamic region. So the next question Dan asked was, how is it that these neurons are driving rhythmic behavior? What is it that they secrete? Now, they're peptidergic, as I told you, so we were particularly interested in peptides that these cells might produce. So we tried to identify these peptides, and I should say that, of course, the first peptide we considered was insulin, because that we knew was expressed here, and it turns out that insulin was not relevant for the control of rest activity or sleep-wake rhythms. So, in fact, the insulin-producing cells even are not required. It's the other cells in this group that are important. So we did single-cell RNA-seq experiments to identify candidate signaling molecules, and then knocked those molecules down by RNAi and tested the flies for effect on rest activity rhythms. And here are some of the peptides that we found expressed in these cells. So they do have insulin-like peptides, and then they have a number of others. And the one we ended up focusing on, or rather Dan ended up focusing on, is this one right here, just called diuretic hormone 44, or DH44. And what DH44 is the Drosophila ortholog of corticotrophin-releasing factor, or CRF. And it turns out that DH44 is expressed in only 6 cells in that PI region, 3 on either side of the midline. And these neurons project down to the ventral part of the fly brain. So so this is DH44 in green here and here. And what this is showing you is that there is a promoter that labels more of these neurons than just DH44. Some of those other neurons are also important for rest activity, and I'll mention them in a minute. Okay, so these cells we found were important for rest activity rhythms. So if you just activate or inhibit or ablate just these 6 neurons using a promoter for DH44, you disrupt rhythmic behavior. Now, that tells you that the cells are important, but what about DH44 itself? And so Dan knocked down DH44 using RNAi, and using 2 different RNAi lines, you see here that expression of DH44, shown here, is totally gone. And when you do that, rest activity rhythms are degraded. So here are control flies, this one right here, and that one right there, and this one at the bottom, and you see they have very robust rest activity rhythms. When you knock down DH44, the phenotypes range from complete loss of rhythms to weak rhythms. So this is actually consistent also with the neuromodulatory effect of peptidergic signaling. In other words, when you get rid of a peptide that's involved in a process, you typically don't have an all or none phenotype. You sometimes weaken, you get weak phenotypes, and that's exactly what we got with DH44. So I'm summarizing a lot of data here, but basically what we found through this work was, as I've told you, that there's a polysynaptic circuit extending from the central clock cells through the DN1 cells to this region called the pars intercerebralis, which is like the fly hypothalamus. And within this region are the insulin-producing cells. And these are 7 cells on either side of the midline. And these cells are not important for rhythmic behavior. But then there are 6 cells, 3 on either side of the midline, that produce DH44, and those are important for rhythmic behavior. And then there are 4 cells that produce yet another peptide, SIF amide. And those 4 neurons are also important for rest activity rhythm. So we're dissecting this region and finding that some of them are involved in rhythmic behavior, and some of them are not, but they all receive circadian input. And it looks like the insulin-producing cells, not surprisingly, have a role in rhythmic metabolism rather than in rhythmic behavior. Now, more recently, we've also found, I told you that these neurons project down to the ventral part of the fly brain, and we found that there is another population of peptidergic neurons secreting a different peptide in the ventral part of the fly brain. And those neurons project directly to the motor neurons to produce activity. So we feel we're pretty close to having the whole circuit, and we started to also record from the PI region and are getting a sense for the type of electrical activity that is displayed by these cells. Now, I should stress that, I'm talking about sleep, and you might have noticed that here, when I'm showing you the data with respect to mapping these neurons, we're really mapping a circadian rhythm of locomotor activity, okay? And that is definitely the case. It's easier to map the cells that drive a rhythm of locomotor activity than a rhythm of sleep, but of course, the rhythm of sleep is intimately connected to the rhythm of locomotor activity. But I will point out that since we published this work, another lab has found that these neurons here, the clock cells DN1s, some of these neurons also have a role in the direct circadian regulation of sleep. So we think it will be the same circuit, and there are going to be, I've oversimplified it, obviously, by telling you it's just this straight linear pathway, but of course it's more complicated than that. There's feedback at many levels, and some of these neurons have tangents, offshoots to sleep-regulating centers. Okay, so that's with respect to the timing of the behavior. How do you get the timing of the sleep-wake cycle? What I've told you is that it requires a polysynaptic circuit from the central clock cells to through the fly hypothalamus, the PI. And within this circuit, the fly CRF or DH44 is required for rhythmic behavior. Now, moving on to the why question, in other words, the function of sleep. This, of course, is a major mystery in biology, right? Why do we sleep? We spend a third of our lives sleeping, and yet we don't know why we do it. And so it's clearly an important question. And most of you are probably aware that the most popular hypothesis for why we sleep is that we consolidate our memory when we're sleeping. And that may be the case. At this point, there's evidence from flies, from mice, from humans, which, for the longest time we called it a hypothesis, and I guess we're still calling it a hypothesis, but I think we can maybe move beyond calling it a hypothesis at this point, because I do think it's supported by a lot of data that memory is consolidated. But consolidation of memory is just a part of a broader process that sleep may be important for, and that is synaptic plasticity. Okay? And a lot of synaptic plasticity in the adults occurs in the form of consolidation of memory. But when individuals or when animals are growing, there is a lot of synaptic plasticity in the nervous system in the course of development. And so it's the young animal synaptic plasticity that we were interested in that I'm going to tell you about a bit. So the idea that sleep may be required in early life is not a new one. So way back, Bill Dement, who's one of the fathers of sleep biology, said in this paper in 1966 that shortly after birth, before there's a lot of stimulation to the nervous system, you have a lot of structural maturation and differentiation of sensory motor neurons, and sleep might help in this type of maturation. Okay, so he had already been speculating that this might be the case. And the truth of the matter is that all young animals sleep a lot. Young flies sleep a lot, as I'll just show you, and young rats sleep a lot, and of course human infants sleep a lot. So I would say that to date, the best evidence for a best example of something that sleep might do in these early stages came from Marcos Frank my former colleague at Penn, and he showed that it's important for plasticity in the developing visual cortex. Now, the paradigm that Marcos Frank was using was one of the ocular dominance. So this basically is the phenomenon in which if you suture one eye in young cats, then the representation of the other eye in the cortex changes, okay, because now you only have visual input from the other eye. And he showed that sleep was important for that plasticity. So Matt Kaiser, a former postdoc who now also has his own lab, recently established, set out to ask the question of whether sleep was required for plasticity in young flies. So just to show you that young flies do indeed sleep a lot, here is the sleep record, kind of like the one I showed you earlier. So now we're looking just at sleep. This is not activity. So sleep over several days averaged here, actually from days 8 to 10. So this is from a day 1 fly in the light bar, and from a day 8 to 10 flies shown in, it's from a population shown in the dark bar. And you see here that the young flies sleep much more, especially during the day. So a lot more sleep. And for those of you who are wondering what a day 1 versus a day 8 to 10 fly means, 1 day in the life of a fly is roughly 1 human year. Okay, so it's a 1-year-old fly compared to a 1-year-old human compared to an 8 to 10-year-old. And not only do these young flies sleep more, but they also fall asleep faster. So the latency to sleep, the time it takes them to fall asleep at lights on or at lights off, is much less than in the 8 to 10-day-old fly. Okay. And the sleep bout duration is also longer. So I'm summarizing a lot of data here just to tell you that Matt, went about asking what the mechanism was that keeps sleep high in young flies. And I'll just summarize this to tell you that it is a reduction in brain dopamine. So brain dopamine is lower in young flies as compared to older ones. And dopamine in flies, as in mammals, is an arousing molecule. So when the flies have less dopamine, they're sleeping more. So Matt went on to ask, is it dopamine generically everywhere, or is there a specific circuit that dopamine is required in that is less active in young flies? And the circuit he ended up focusing on is one shown here. So what you're looking at here is you're looking down on the fly head. Here are the compound eyes, and the various structures of the brain have been put in as a schematic on top here. And the structure I'd like you to focus on is this red one right there, and that is the dorsal fan-shaped body. And now Paul Shaw had shown that the dorsal fan-shaped body is a sleep-promoting structure in the fly brain. And Mark Wu's lab and Kazuhiko Kumei's labs showed that this fan-shaped body receives dopaminergic input. So typically, during periods of arousal, the dopamine input to the fan-shaped body inhibits sleep and promotes arousal. This is one of the mechanisms by which dopamine promotes arousal. But then during periods of sleep, the fan-shaped body dominates. And what Matt found was that the dopaminergic neurons projecting to the dorsal fan-shaped body are less active in young flies. So what he did was to use a calcium indicator, Calexa, to monitor neural activity in the dorsal fan-shaped body in young flies, day 1 flies versus day 7 flies. And you see here that the activity of these dopaminergic projections is less in day 1 and greater in day 7. So the dorsal fan-shaped body then is more active here and promoting more sleep than here. And the quantitation of these data are shown here. So on day 1, the dopaminergic neurons are less active. Now, just to show you that it's not universally true that all areas of the fly brain are less active in young flies, here's another region which is actually more active in young flies, and those data are shown there. So we know the circuit then that maintains high levels of sleep in young flies. But why do you need that sleep to be high? Or does it even need to be high? And it actually does need to be high because it turns out that Paul Shaw had shown earlier that if you deprive young flies of sleep, then their courtship in later life is impaired. And now male flies go through this very elaborate courtship ritual, which is shown here, where when they're courting a female, they put it the female, a virgin, the male fly will orient towards her, he'll start tapping her, he'll sing, which basically means flapping his wing here, he'll lick, and then he'll attempt copulation, and then at the end, they'll copulate. So this courtship behavior is disrupted if flies are deprived of sleep very young, the male flies. So Matt asked if he could get the same effect on courtship, not by mechanically depriving flies of sleep when they're young, which is what Paul Shaw had done, but through stimulating the specific dopamine neurons he was looking at. And so the paradigm is to take very young flies, stimulate the dopamine neurons for a day and a half, shown in the pink, and then test them 3 days later. And then do the same thing with day 7 flies, so in other words, stimulate the dopamine neurons and test them 3 days later. And what he found was that sleep loss through stimulation of these neurons reduces courtship. So courtship index is the amount of time that the males spend courting. And the courtship index is reduced when they were deprived at day 1 and then tested at day 5. When they were deprived at day 7 or 8 and tested at day 12, courtship index is not affected. So sleep depriving the mature male doesn't impact courtship. Now, what was even more dramatic than the amount of time spent courting was the copulation percentage, which is greatly reduced in the young flies. So what that says is that even those males who started to court could not maintain the courtship to see it through to completion. In other words, to copulation. And I'm going to show you a video here where, okay, so on the left is a male that was the control male, and he's courting a female. On the right is a sleep-deprived male, sleep-deprived in early life. So he's now being tested for his ability to court 3 days later, and he's also courting a virgin. Neither of the females was sleep-deprived, only the male on the right was. And so you see here that in this time I've been talking, the male on the left has gone through his courtship ritual and is already copulating. And the one here on the right is chasing after the female. He's flapping his wing, so he's going through some of the steps of courtship, but he somehow keeps losing it. So he gets distracted, there he is, and then he resumes again, starts the ritual again. And so it's not like, he's keeling over and, passing out or something. He just doesn't seem to get his act together. And you'll see at the end of the video, there he is. So that's actually the typical response of a male that was deprived in young life of sleep. So we know how sleep is kept high in young flies, and we know that it is required for adult courtship. But what is it that the sleep does in the young flies that impacts courtship later? So our hypothesis, of course, was that it's affecting courtship circuitry. So the question we asked is, are specific circuits affected by sleep loss in early life? And we knew that the olfactory system is very important for courtship because flies need to sense odors and pheromones for courtship. And the fly olfactory system is similar to the mammalian. There are olfactory sensory neurons that project to these glomeruli in the antennal lobe, and the glomeruli then, through projection neurons, go to higher order structures. So Matt asked if anything in this system was impacted by sleep loss. So the protocol is the same: sleep deprived by stimulating dopamine neurons in early life, test 3 days later, in later life, and test 3 days later. But instead of looking at courtship behavior now, he's dissecting the brains and looking at olfactory glomeruli. And he's looking, I'm showing you data from 4 olfactory glomeruli marked here, and they're shown here. And then the same thing is done in the sleep-deprived animals. And what he found is that this one particular olfactory glomerulus, VA1V, is reduced in growth when the animals are sleep-deprived. So all the other glomeruli are the same in control and sleep-deprived animals, deprived at day 5 or day 12. VA1V growth is reduced in animals that were deprived when young. So why VA1V? Why is this particular glomerulus affected? And we asked, could it be that sleep loss causes shrinkage of this glomerulus? Or is it that this glomerulus is normally growing a lot in early life, and what sleep loss does is to stunt the growth? And the answer is that this is an actively growing glomerulus. So post-hatching of the fly, this glomerulus shows the most growth. So all of them, these are 4 different glomeruli that were tested, all of them show some growth from very early life up to 12 days. But this one grows the most. And you can see that here from 2 to 3 hours to 12 days. So we believe this is most susceptible to sleep loss because it is the most actively growing glomerulus in early life. And so what that says is that in early life, the parts of the nervous system that are growing the most or changing the most are the ones that may need the most sleep. So just to conclude this part then, what I've told you is that we've addressed the function for sleep in early life, and we find that the sleep is kept high in young flies by downregulation of dopamine in a specific circuit. And that downregulation of dopamine allows high activity of a sleep-promoting structure. And that sleep is required in early life for development of courtship circuitry. Okay. So in the final part of my talk, what I'd like to talk about is this homeostatic control of sleep, regulation of sleep onset and maintenance. Now, I mentioned that when you're looking at homeostatic control, it's really the drive to sleep that you're measuring. Not the timing, it's the drive to sleep, so which sometimes can occur against your clock. If you've been up all night, you will sleep in the morning, although at that point your clock is telling you to wake up. And so that is your homeostatic system at work. So when we're looking at the homeostatic control of sleep, what we're usually measuring is duration of sleep and sleep intensity. Now, maybe you've already gathered this, but we can't do EEGs in flies. I should say that Ralph Greenspan's lab has done electrophysiology on sleeping and awake flies and validated our definition of sleep through electrophysiological methods. But electrophysiology is not something we can routinely do to measure sleep, so we use our behavioral assays. And in those behavioral assays, when we look at circadian control, we're looking at when sleep occurs. When we're looking at homeostatic control, we look at sleep duration and sleep intensity. So the major neurotransmitters, in terms of how they affect sleep duration or intensity, actually have very corresponding effects in flies and mammals. And so dopamine, norepinephrine, serotonin. The work on serotonin and sleep was actually done by Kuan Yan who's an investigator here now and was a graduate student in my lab. And histamine, GABA, they have the same effects. But we didn't develop a fly model for sleep to show that what is true in the mammal is true in the fly. I mean, the reason you do it is because you want to go beyond that and you want to find new things. And how do you identify novel sleep genes? So the unbiased approach, of course, is the best approach. And I feel like that's particularly the case when you're studying something like sleep about which we know nothing or very little. The unbiased approach, or forward genetics in this case, where you mutagenize animals, screen them for phenotypes, and then figure out which genes were mutated to give you less sleep or more sleep. I have a bias, but I feel like that approach really lets the animal tell you what's required for your process of interest without you coming up with any preconceived notions. And the fly really lends itself beautifully to forward genetics. We know it's well established as a circadian model. As a genetic model, you can screen lots of flies. You can go through rapid generation times and so forth. There's also a really important proof of principle here, which is very relevant to the study of sleep, and that is that the whole circadian clock mechanism was figured out in Drosophila. So circadian genes were identified in Drosophila through such forward genetics, and these genes are mutated now, we know, in human circadian disorders. Okay, so we had this great example of how the fly had been a good system to study circadian rhythms. And I would make the case that it actually has an even more of an advantage for sleep. And the reason is that sleep is an essential process. And so a loss of sleep is very easily compensated for. So we find, for instance, when we get a mutant that affects sleep amount, if you maintain that mutant as a homozygote, over time the phenotype gets weaker. Because I think they pick up modifiers. And that just tells you that the animal tries to compensate through redundant mechanisms or otherwise for a loss of sleep. But this type of redundancy and compensation, even though we do see it in the fly, is much less than what you would have in the mouse. So it's very hard to get very strong sleep phenotypes in the mouse, but in the fly you can get them, and I'll show you that in just a minute. And so we've been using forward genetic screens. Susan Harbison, who's an investigator here now, was doing forward genetic screens, and she was a postdoc in my lab and is continuing to work on the genetics of Drosophila sleep. So I went backwards. So here's an example of this very dramatic, robust phenotype you can get in flies. So here is flies in sleep in male flies and in female flies, shown in the dark circles, the top and the bottom. And then here is the first mutant we got from a genetic screen in flies for mutants that sleep less. And we called it sleepless. As George said, we can give our flies cool names. So the sleepless mutant, you can see here, sleeps a lot less in males and females. So sleep is reduced by like 80%. So now we and others have done a number of screens for altered sleep duration. And shown here are many of these genes. Okay, so they affect voltage-gated potassium channels, dopamine signaling, GABA signaling, cholinergic signaling. And so the one thing that I guess strikes one when you look at this is, so it's the same old neurotransmitter pathways. This is not like the circadian system where we were finding dedicated clock genes whose only function it was to drive a circadian rhythm. These are the neurotransmitters that are required for a lot of things. And so I will just suggest that maybe it is the case that baseline sleep or normal daily sleep amount is really controlled by these known neuromodulators. Okay. The important point, and for this I will highlight two of these mutants here. One of them is Shaker, which affects voltage-gated potassium channels, isolated by Chiara Cirelli and Giulia Tononi. And Sleepless, which is a small GPI-anchored protein that regulates Shaker and nicotinic acetylcholine receptors. They're hitting these two pathways, right? Voltage-gated potassium channels and nicotinic acetylcholine receptors. And the same ones are implicated in studies or investigation of genes underlying human sleep. So these genes were found through unbiased approaches in Drosophila, and they are implicated in human sleep. So the first paper here is referring to this collection of neurological disorders where these people have neurological symptoms, but they also have insomnia. The best example of this is actually Morvan syndrome. Turns out that these people have autoantibodies to voltage-gated potassium channels. Okay, and then the other example comes from a huge GWAS study carried out by a European Union collaborative group where they looked for SNPs affecting human sleep duration. And they focused on a KATP channel, which affects sleep in humans, and they showed also in flies. But what was really interesting to us also about this study was that among their top hits were a nicotinic acetylcholine receptor alpha subunit, which also came out of our fly screens, and a regulatory subunit of the shaker voltage-gated potassium channel. So even when you look for genes affecting human sleep duration, it is the same ones that we're finding through unbiased screens in Drosophila. So I'll put it to you that maybe these are the neuromodulators that affect normal daily sleep. So what about sleep homeostasis? How about when you don't sleep at night and you sleep during the day, and so you're really responding to homeostatic pressure? Are those genes different? And so we've actually done a screen for altered rebound, where we just deprive the flies, don't let them sleep at night, and then they have a rebound in the morning, and we look for ones that have altered rebound. And we basically are finding that while we got rebound mutants at a very low rate, I might add, in fact, sleep genes in general we're not finding at very high rates for robust phenotypes. We get about 1 hit per 1,000. But when we look at the 2 rebound mutants that we got, we cannot map the effect to single genes. So it most likely is a polygenic effect. It may be the case that it's very hard to get a single gene that can do away with the need or the manifestation of a sleep rebound. So I'm not going to talk about that screen, but I just wanted to point that out. And then we started asking, well, okay, so we're doing loss of function screens, right? And so when we do loss of function screens, we get altered sleep duration. Now, I should say, too, that when we do loss of function sleep screens and we get mutants that produce less sleep, then our hope was that when we took those genes and overexpressed them, that we would now get high sleep. And we didn't. Not in any case, okay? So again, you're kind of left with, all right, so these genes are permissive for sleep. They need to be there for normal sleep amount, but they are not instructive. Or at least by themselves, they're not instructive. You cannot overexpress them and drive sleep. So are there any genes you can actually do that with? Can you induce sleep? And that work was done by a current postdoc, Hirofumi Toda. And what he did was an overexpression screen. And what he basically he used the GAL4 UAS system. For those of you who are not familiar with it, GAL4 is a transcription factor, and it's usually expressed in a specific pattern in a fly. You can cross that fly to other flies that contain a target gene that is driven by a sequence recognized by GAL4. So the pattern line, or the pattern GAL4 that Hiro used for his screen was a pan-neuronal one, which is induced by RU486, because we wanted something that we just turn on in the adult and then look at the effect on sleep, see if it induces or reduces sleep. And so he took this and he crossed this pan-neuronal one to 12,000 different fly lines with different target genes flanked by the GAL4 binding site. And so, yeah, to be precise, 12,348 lines. And the results are shown here. So when you turn on the particular gene that's in each of these lines with this inducible GAL4, you get this wide distribution. Sometimes you get an increase in sleep. Sometimes you get a decrease. This here is a control. We know that this particular line dramatically reduces sleep. And then so what Hiro did was to pick the ones in red and further screen them. So he screened them and rescreened them and figured out whether they were the same gene or different genes. And as a result of all of this, found 5 lines. So just 5 lines, which represent 4 genes out of 12,000. And 3 genes produce short sleep. And they are a GPCR, a guanine nucleotide exchange factor, and a serine type endopeptidase. And so we're characterizing these right now to figure out how it is that they reduce sleep. We're getting loss of function mutants, and we hope that the loss of function mutants will show high sleep, but that's not always turning out to be the case. Sometimes even the loss of function has less sleep. But the one that was more interesting to us was the one that induces sleep. So this is only 1 out of 12,000. And it's unknown. So the biochemical function is unknown. We're calling this nemuri, which actually means sleep in Japanese. And it has a signal sequence, and it's small molecules. So we think it's a secreted factor, but we haven't demonstrated that yet. It may be a peptide. I actually showed it to somebody who's very into peptides, and this person seemed to think it was a peptide. And it's got a lot of arginines and glycines. And so here are the data. If you overexpress this, you induce sleep in flies. So here are male and female. So blue is the uninduced male and the uninduced female. And then when you induce expression of this, you increase sleep. It's actually very dramatic and very reliable. Doesn't even require that much induction. And this gene, it turns out, is normally induced in response to stress. So we think that what it might be doing is increasing sleep under those conditions. So just to conclude there, what I've told you is that when you look at, as far as regulation of sleep onset and maintenance, those that affect daily sleep duration map to the known neuromodulators and regulators of neural activity. And this is true in flies. It's also true in humans. Other sleep phenotypes, such as altered rebound or just sleep induction, are really hard to get positives from genetic screens. And I'm speculating that this may be because most of the time it's hard to get a single gene that actually affects it. Okay, so I'd like to conclude then, just acknowledge the people who did the work. The mapping of the circadian circuit was done by Dan Cavanaugh. The sleep function in early life was Matt Kaiser, both of whom now have their own labs. Hiro Toda is currently in the lab, and he is doing the overexpression screen. Jifeng Yu helped Matt with the early sleep experiments. And I just wanted to point out, this is actually an old picture, and Shailesh Kumar, who's now a staff scientist here, was in my lab at the time. I didn't get a chance to talk about his work. And I'll stop there and be happy to take questions. Thank you.
Very exciting talk, and I have a question about the different possible differences between daytime and nighttime sleep. One of your earlier slides showed that the lengthening of sleep time in the one-day-old versus the older flies is almost entirely during the daytime. So is there something to this expression that daydreaming helps forming memories and so on?
Oh, that's a great term for that, yes. So daytime and nighttime sleep are different. They're different in terms of sleep intensity. So it's easier to wake the sleeping fly up during the day than it is at night. The arousal threshold is different. Why it is that daytime sleep is higher in the young fly, we don't know. I mean, one possibility, that's a really intriguing possibility, like I said, that it's daydreaming. We always thought that it may have to do with the ceiling effect, that at night there's already high sleep, so it's hard to push it anymore, whereas during the daytime there's a lot of room to grow. But yeah, I don't really know. But it also is the case that, so in the mature flies, the daytime sleep is light sleep, so they can be easily woken up. In the young flies, it's actually not. It's very hard to wake them up.
So I was just wondering, with the young flies, if you noticed any changes in eye morphology, because one of the hypotheses of the purpose of sleep is that it has something to do with eye development in developing infants.
Is it eye development or is it visual system in general? Do you know?
I think both.
Yeah.
Like the optic nerve development, for example.
Yeah, so we have definitely, so the short answer is we haven't noticed anything morphologically about the eye. But we've been intrigued by, data that have been published that say that there's plasticity in the visual system. So obviously I mentioned the cat stuff there is, but even in the fly, there's plasticity in the visual system and in the olfactory system in early life. So you can actually, modify their responses with different types of behaviors or different types of environmental paradigms, exposing them to constant light and so forth. We haven't really tinkered with that. So I think that the structurally, I don't think there's a difference, but it's possible that the visual system is still developing and requires sleep.
I just had one other small question, which is whether or not you think Numuri might be a biomarker for sleep, like it might increase and decrease with sleep.
I'm sorry, whether Numuri might be a biomarker.
Right.
We're building reporters for it now and testing that out, yeah.
You've discussed the chronobiology of sleep,
Right.
but chronobiology is also significant in pathologic conditions,
Yeah.
such as asthma and characteristic febrile curves and diseases such as tuberculosis.
Right.
Do you have animal models as such for any of these entities in order to explain these type of phenomena, which would be of particular interest, not so much in febrile curves, but would be of interest in asthma?
Right, so definitely not in the fly. There are mouse models. I don't know if they've looked at circadian regulation. It is, what has been looked at, I can tell you, is in the normal animal, there is a clock in the lung, and the macrophages there have a clock and have a huge role in it. There's a rhythm of mucus production in response to inputs through the vagal nerve. How that's relevant to asthma, I don't yet know, but of course it's also known that asthma attacks tend to occur at specific times of day. With respect to the models you're talking about, the only model, the models that we've actually spent some time studying are neurofibromatosis and Fragile X, and they actually have circadian and sleep defects.
Okay, thank you.
Very nice talk. Thanks, Amita. I just wanted to ask a question about sort of the idea of sleep and sleep deprivation in young animals. I mean, you were talking about flies at day one, and yet, of course, fly has already undergone 10 days of development prior to that.
Yes.
And so, in a sense, the developing animal is, the larva. And I'm just wondering if your lab or any other lab has looked at rest activity rhythms in larvae and whether or not the genes that affect the sleep-wake rhythms in adults are also affecting the activity rhythms in younger flies.
You should ask Susan about that, actually. So a couple of my former postdocs, Matt Kaiser even, and Susan, are looking at larvae to see whether there is a sleep-like state. I would say, though, that while you're totally right that most of development is occurring in that 10-day period where it's going through egg and larval, I think there's still development occurring in the hatched fly, just as there is in the human infant. So sort of in my naive world, the 10 days of the fly egg to larva is, to me, analogous to the development of the fetus in utero. And, the baby's born, and there's still stuff happening, which is what we think we're looking at here. Any other questions? All right, thank you.